Hello, and welcome to this webinar introducing Benchling's new capabilities to support RNA therapeutics. We are thrilled to have you here with us today. I want to start by acknowledging that the attendees in this discussion represent a very select group of scientists. We believe that those of you working with modified oligos are at the bleeding edge of research, fundamentally shifting how we can treat diseases. And Benchling is frankly honored to be able to support your endeavors in this space this week onward. So we want to thank you already for being part of the conversation and early work unlocking the power of RNA therapeutics. My name is Kate Quigley, and I'm a product marketing lead at Benchling. Today marks our public launch of our RNA Therapeutics R&D solution, which we have been creating for this release. Joining me is Andrew Guo, lead product manager for the creation of the capabilities to support hybrid biologic and chemical molecules. Andrew, could you introduce yourself and tell us a bit about how and why Benchling is eager to support RNA therapeutics research?
Sure thing, Kate. So my name is Andrew Guo. I'm the product manager responsible for our molecular biology and chemistry products here at Benchling. My background is more on the bioinformatics side. Prior to Benchling, I've worked on a variety of different NGS software solutions spanning the clinical diagnostic space for ultra-rare diseases to target discovery and data mining applications for pharma. Throughout my time here at Benchling, we've seen a number of customers who are working on oligo-based drugs targeting RNA that often include increasingly complex patterns of chemical modifications. We've noticed time and time again that there really is an area of unmet need here for customers, as they're almost always forced to design and represent the complexity of chemical modifications in a tool like Excel. And that's really what led us to where we are today. And before I go in at length about the product, I want to just turn it back to Kate to walk through our agenda and set the stage.
So to begin today's discussion, I will talk about the current state of the RNA therapeutics industry, including tremendous gains that have been made and additional opportunities to take advantage of. Andrew will join us again to take us through Benchling's new product capabilities built for modified oligos. And then we'll wrap up with Q&A, which we will be happy to stay on for as long as is helpful. Before diving into the capabilities for Benchling's solution in relation to RNA therapeutics specifically, I want to take a moment to introduce Benchling generally for those who are just hearing of our life sciences R&D cloud for the first time. Benchling was founded in 2012 by a team of scientists and engineers at MIT who were frustrated with the poor quality of software they had available to support their science. With over 300,000 users across the globe, Benchling is now a widely adopted cloud solution that gives users and management a 360-degree view of R&D innovation. We offer 7 integrated applications in a single interface, and these applications span the R&D process from experimental design, in silico biology, sample tracking, and process management. These applications are built on a common platform and configurable data model to support each team's unique requirements. We think that every company and scientific team deserves a solution that will accelerate and support their innovations. To that end, Benchling has been designed with 4 core attributes. First, it has been built for complex science. With our platform, you can readily model the science you focus on, which means having the data model built fit for purpose, as we will see with the modified oligo capabilities today. Benchling also adapts to your process. It supports the processes and workflows that teams perform and has the ability to scale, iterate, and evolve over time. It also enables key decisions by providing users with contextually relevant data at every level, giving scientists insights about their experiments and team leaders data on their programs, so you can make both scientific and operational decisions quickly with complete and accurate data. Lastly, it is easy and intuitive to use. We regularly hear feedback from users how they find value from using the solution immediately, from how it saves them time, and it also adopts in a way that will help them keep compliant and consistent with the rest of the R&D organization. The flexible nature of Benchling allows us to continually expand the types of emerging scientific modalities we can natively support with purpose-built capabilities. We're entering a new era of science where there is no longer just a finite number of validated targets. Instead, we see approaches like bispecifics and ADCs that can increase the druggable population of targets. We're able to focus on bringing together biology and chemistry, for example, into an integrated platform, supporting the hybridization of small molecules and biological experiments and assays. The history of biopharmaceuticals is long and yet also rapidly accelerating. Today, modalities are not confined to traditional categorizations as we enter a new era of science where it's no longer just small molecule or antibodies. This opens up territory for targeting previously undruggable diseases, which is a massive opportunity for humanity. Recently, the discoveries in these complex therapeutic categories, including siRNA, miRNA, and ASOs, have been remarkable for their ingenuity and efficacy. By combining the powers of biology and chemistry, RNA therapeutics are the primary drivers of the shift from one drug, one target to multidimensional and iterative targeting. And we've already learned so much about the chemical impact on biomolecules. Scientists have learned to modify every part of the RNA molecule, from the sugar to the base to the linkages and conjugates. There are now some well-known chemical modifications that are in frequent use today across industry, and these have had dramatic impacts on how successful the RNA will be. With this greater understanding of the impact of the chemical modifications, tremendous improvements have been made in recent years to increase the potency, stability, durability, and precision of these biomolecules. Yet few get to rest on their laurels, as this work is still underway with additional hurdles to overcome. This iterative rapid discovery process includes increasingly more modifications of a greater variety. To better understand the impact of these modifications, we need to be able to track, systematize, and connect chemical design to biological assay context within a centralized data model. Additionally, there's a historic lack of a standardized language or syntax to represent chemical modifications on nucleotide sequences. Research teams spend a significant amount of time translating their bases into shared language for easier readability. Together, a centralized data model and standardized nomenclature will significantly accelerate time to insights and treatment delivery to market. As these approaches have gained traction scientifically and clinically, the tools to engineer them have been frustratingly limited, however. Scientists chemically modifying RNA oligos have had to contend with manual, low throughput, and disconnected software and spreadsheets to complete the full workflow of designing, analyzing, and testing these complex biomolecules. To date, there has not been a solution that unifies the biological and chemical nature of RNA therapeutics with seamless collaboration, analysis, and scientific insights. Data is disconnected by living across multiple systems, so contextual insights are never surfaced. Time is wasted transferring data, and the process is error-prone. This current ecosystem is not efficient or scalable. Solutions that focus primarily on chemistry workflows also struggle to model hybrid chemical modifications to large biomolecules. And solutions that use the commonly available open Helm editor are also not intuitive for biologists. Given that there's also significant partnership opportunities for many RNA therapeutics companies, it is also important to highlight that data sets with academic, industry, and CRO partners will be siloed and extremely difficult to control shared access to and connect with when the time comes. By contrast, Benchling is built on a modern, unified cloud infrastructure that provides a configurable data model that maps to the team's unique scientific and operational requirements. Having a unified data model is critical to be able to ensure the full traceability of samples and experiments, as well as the ability to perform ad hoc and systematic queries of the data for analysis, samples, and process intelligence. All of the Benchling applications are built and delivered through the Benchling platform, which powers user interface components and provides infrastructure that delivers security, scalability, and integration capabilities. We know that most companies have a rich ecosystem of instruments, software applications, and data stores that are optimized when data flows freely to support key R&D functions, including preparing IP filings and IND submissions. So Benchling provides a full set of REST APIs and out-of-the-box connections for lab automation, as well as ways to move data onto and out of the platform. So the RNA therapeutics capabilities Andrew is about to share with you are built on this foundation and extend to support the specific needs of modifying oligos. Andrew, could you walk us through those capabilities?
Sure, Kate. So I'm proud to announce that scientists can now design, analyze, and develop their RNA therapeutics directly on Benchling's unified informatics solution. We've built out support for Helm into Benchling's flexible data model to help standardize the representation of chemically modified nucleotides. And we provide a standard set of common chemical modifications like phosphorothioate or 2-O-methylribose, but it can also support custom modifications in our monomer library. This allows scientists to design and track oligos with complex patterns of chemically modified nucleotides directly within Benchling in a standardized fashion. As with all data captured in Benchling, the aim is to really increase the ability of scientists to accurately keep track of all of their underlying data assets, as well as any associated experimental results in a highly structured manner to ensure high data integrity. Teams can now automatically centralize all of their oligo-relevant data, such as inventory locations, sequence history, and any pertinent experimental or analytical results. And this improves data traceability and makes it easy to ensure that all work performed, no matter by whom on your team, can all be linked to the same underlying data entities. The platform can also import, register, and create different modified oligos and perform the sort of uniqueness checking to make sure that any duplicate sequences are flagged and the team isn't spending any time working on redundant data objects. For the first time, scientists can now design chemically modified oligos, standardize on syntax, centralize experimental results, and collaborate with teammates on a single platform. With Benchling, researchers can create cutting-edge RNA therapeutics and bring these incredible therapies to market faster. So with that, I'm going to actually switch over to a live demo and give you a quick preview of all of the functionality that we have built out. So just really quickly for folks who are new to the Benchling platform, this is the Benchling web application. Benchling's a cloud-based platform. I've signed in here, and immediately on the left-hand side here, you can see the Benchling home page, projects, search tool, as well as all of the other applications that you might be able to bundle into Benchling, such as our registry, inventory, workflows, and requests tool. Right now, I'm actually in a Benchling notebook entry, and so I just want to quickly walk through a few concepts and show you what we've built. So first, I mentioned that we have built out support for Helm, directly went into Benchling. And this means that we now have a standardized way of representing any sort of chemical modification on nucleotides. So in Helm, they actually define 3 different subtypes of modifications, and they're called monomers. Each of the component monomers that you can customize basically fall into either being a phosphate linker derivative, a ribose sugar derivative, or a nitrogenous base derivative. To be able to support building out custom chemical modifications directly within Benchling, we've introduced a concept called a monomer library within Benchling. And this is an item that lives within our registry product. So our monomer library, which I'm going to just quickly run to right now, is actually a store of all of the chemical modifications, all of the custom modified, riboses, modified phosphates, or modified nitrogenous bases that are available in a customer's environment to be able to be integrated into any of these modified oligo entities. And so you'll see here a set of very common modifications, ribose modifications like 2-fluororibose, 2-O-methoxyribose, et cetera, as well as some modified phosphates like phosphorothioate and a modified nitrogenous base, 5-methylcytosine. So once a custom modification has been specified in a monomer library, they immediately become accessible and usable in any sort of oligo entity in Benchling today. They can immediately be integrated into any existing piece of data. And so let me just hop back here and import some data. Something else that we've added support for in addition to Helm, though we recognize that Helm is a great standard notation for keeping track and basically being the underlying data model for representing all of these modifications on oligos, we recognize that a lot of different customers are using different vendors to synthesize and create these oligos. And so as a part of rolling out our initial support for modified oligos, we've actually also added support for IDT syntax. And so here I actually have a few oligos specified using IDT notation. So if I just copy this, I'm able to directly go in and import a set of oligos. Let me just paste them in here. You'll notice that we automatically detect that the syntax that was used is actually IDT notation here, and that comes preset. Obviously, you can also specify oligos using just the natural bases the ACTGs use, or Helm notation if that is something that you guys are currently using. In this case, my oligos have come formatted in IDT, so I'm just going to go ahead and import them. No errors upon import, and so I will go ahead and open some of these oligos. You'll notice here that I'm actually in a project view now, and so I've within Benchling there's a basic folder structure of projects and folders that lets you both manage permissions but also compartmentalize your data into specific programs or projects, however you see fit. In this case, I have a single folder called Modified Oligos that I've uploaded these 3 new oligos into just now. So if I open up a single oligo here, I want to spend a little bit of time walking you through this particular UI. And so what we've actually added here is a new visualization framework for representing and visualizing these modified oligos. So you'll see here a sequence map for this oligo entity and a few interesting kind of demarcations on the first 3 and last 3 nucleotides. If I hover over any of these nucleotides, I'll actually get a view that describes the particular sugar monomer, the particular phosphate monomer, and the particular base monomer that was used at this position. In this case, the first adenine here includes 2-O-methylribose and phosphorothioate. And this is indicated by the circle around the A, which is meant to represent modified sugars, and the diamond shape here, which is meant to represent any sort of modified phosphate. You'll see in the legend down below that I can actually hover over all of the modified sugars here, which will highlight on the sequence map where which nucleotide positions actually have a modified sugar included. Same for modified bases, so I haven't integrated any. And then the same thing for modified phosphates. So this basically tells me that the first 3 and the last 3 positions have both modified sugars and modified phosphates. If I were to want to edit any of these nucleotides, we've introduced a new nucleotides table shown here to allow you to basically make point-by-point edits across your oligo. And so this means that if I wanted to alter the particular sugar monomer used at any given position, I can hover over any of these rows. you'll notice the same sort of indication on the sequence map that tries to steer you towards exactly which position you're introducing an edit in. And then you can just simply click in, and you'll see all of the potential sugar monomers that were previously shown and specified in your particular monomer library. So in this case, I could choose to, alter this first 2-methylribose to 2-fluororibose or what have you, and continue making edits in that fashion. Outside of the nucleotides tab, we do also have a few other key enhancements on this view that I want to just highlight. The other is with version history. And so with any sequence entity in Benchling, we keep a robust version history of every edit and every state that sequence entity has been in. You can kind of think of it like analogous to a Google Doc version history. So you'll see here, because I haven't introduced any edits, I just have the original sequence changed basically exactly when I imported. We'll come back to this later after introducing some edits. The other pieces that I'll just briefly highlight are, obviously we have, because this oligo entity is, an entity in Benchling, you can always attribute different results data directly to this entity. Any results that are introduced and linked to this entity via a notebook entry will be aggregated and shown in the results tab. If you wanted to add in any sort of free text description for this entity, you can do that in the description tab. And what I will spend a little bit of time on is the metadata tab. Within the metadata tab, we allow you to specify specific fields of metadata that are important for your experiment or for your specific project to track against this entity. And so within our registry, you can actually customize what we call data schemas. They're essentially pre-configured sets of fields of data that need to be captured for each of these entities. And so I've actually configured a schema already, just simply called modified oligos. And this schema that I've configured actually has 2 fields of data that are actually automatically computed and populated by Benchling. And so if I assign this schema, you'll notice that there are 2 function or computed fields. One to produce the Helm notation and one to produce the IDT notation form of this particular oligo. So this will always dynamically refresh and basically represent the current state of modifications in your oligo. If you introduced new monomer changes to this oligo, the Helm and IDT notation would change to match what is shown in the sequence map. So in the case that you needed to export or copy out this information and send it into a different location, that's always easy to do. After setting a schema for an entity, we can actually now register this entity and formalize it in Benchling's registry. Our registry is essentially a lockdown version of a database for each customer, and you'll see here that I have a few different types of data already registered in this particular registry. Some demo compound cell lines and gene constructs. In this case, I've actually already registered this exact oligo with this exact pattern of modifications. And so you'll see that here marked as the same name, oligo 222, but underscore duplicate. And so one big change that we made is to our registry uniqueness constraint feature, which is a way to basically flag duplicate data entities that a user might attempt to register. In cases where a duplicate is identified, we will flag that to the user and basically ask if they actually meant to merge or create a lot or batch child entity against that previously registered item. And so you'll see that if I actually go through the process of trying to register this entity now, the loading screen actually is a live check against the registry. And so you'll see here that a duplicate error was identified, saying that the sequence bases match exactly an already registered entity. So if I continue to move forward here, these items would be merged together, meaning that any data that was attributed to one or the other entity would be merged and combined together. This is really helpful in cases where you have a large team collaborating on the same sets of data. This ensures that you're not actually tracking your results or analytical data against two separate entities that are in fact identical and should be merged together and combined. So in this case, we've actually made our registry uniqueness constraints not just nucleotide base aware, but also chemical modification and position aware. And so this means previously, if I was to register the exact same nucleotide sequence, but with a different modification pattern, those two entities would be considered duplicates because their bases match exactly. In reality, you might have the exact same natural base sequence, but with n number of different modification patterns applied, and you would consider all of these to be unique. So in this case, I'm actually going to go back into the nucleotides tab here, and I'm going to make an edit, but I'm not going to make an edit to any of the sequence bases here. I'm going to keep all of the bases identical. I'm actually also going to keep the positions of modifications identical as well. And I'm simply just going to change the modification that I have introduced on the first nucleotide here from 2-O-methylribose to 2-fluororibose. You'll see now that I've introduced this change on the nucleotides tab. The Helm notation has adjusted to use the 2-fluororibose monomer encoding, and the IDT notation computed field has changed to use the IDT modification code for 2-fluororibose. If I hover over the A now, you'll notice that I am in fact, using this 2-fluororibose at this position, not 2-O-methylribose, and I've not made any other changes to this sequence. If I go over to the version history now, I'll see that the particular change is represented here. Now I have a note here that I've updated a chemical monomer at this position. If I choose to revert back to the original sequence, I can click in and I can see the original state at this time point of edits. If I wanted to reset to this original version, I could always do that. And this version history is a standard feature of all sequence entities in Benchling today. You always keep track of every edit that is introduced to an entity, a particular user that has performed it, and a particular timestamp at which the action has been made. So let me actually not revert back, but once I have introduced this edit, I'm now going to try to register this entity again. You'll notice that I don't run into any sort of duplicate clashes now. Now the registry is telling me that, hey, this entity with the 2-fluororibose at the first position is now unique and is totally fine to continue with registration. So I'll just go ahead and register that item. So on top of all of the functionality I've showed here with the new visualization tools, the new ways of tracking version history and uniqueness constraints that are all now position and modification aware. All of the standard functionality that you're used to within Benchling it also will apply to these modified oligo entities, meaning that if you wanted to, for example, collect results, associate other entities and create entity links in schema fields, all of that would be accessible to these entities. If you wanted to search for these entities using either Helm or natural-based syntax, you can perform those sorts of searches within our search framework as well. So you'll notice a few different options here to search entities by the Helm string or by nucleotide string. All of this, awareness of chemical modifications has basically been inherited and built out throughout the platform. So with that, I just want to turn it back to some slides to round us out before taking some questions. So let me just walk forward a little bit and go back into presenter mode. Great. So just as RNA Therapeutics has become this new emerging scientific technology, this really requires a new data management and collaboration tool to help with the success of the early development through testing. Benchling really simplifies the collaboration aspect, improves data quality, and provides real-time insights and ultimately accelerate the speed of work for researchers and developers. Benchling is one single connected solution, purpose-built to support all of the complexities of your work in both biology and chemistry. And now, again, multidisciplinary teams can all work with a centralized, with that centralized experimental context to get real-time scientific insights. We also, again, believe that standardizing on a single notation, in this case Helm, will really ease the frustrations associated with translating and analyzing differences that might come from internal or different vendor teams. So with that, I'll pass it back to Kate.
I wanted to mention that we are here to support and stay ahead of R&D needs for this modality, as we are with all of our modalities. And so as much as these are the core values across all of Benchling's capabilities, it's essential in our work with RNA therapeutics companies in particular to note how science-driven, customer-centric, and growth-oriented we are in support of your particularly dynamic space. Benchling created this functionality in partnership with researchers focused on RNA to deliver on the specific needs of this hybrid modality. We will continue to stay ahead of the needs and evolve the product as the science itself continues to transform. We want to mention to our current customers who use our molecular biology application, these RNA therapeutics capabilities are already available to your organization. As a next step, you can simply reach out to your customer support representative and they'll be able to toggle on access to these capabilities immediately. Before we jump into the Q&A discussion, and I appreciate that we've already got a good number in the chat box, we encourage you to learn more about what we're offering on our website. Within the Solutions tab of the navigation, there's a page for RNA Therapeutics. If you would like to speak with Andrew and me directly, please reach out via email. Our addresses are here. I'm kate.quigley@benchling.com, and Andrew is andrew@benchling.com. We're happy to correspond that way as well. We'll also send a follow-up email to everyone who registered, and it will contain a copy of this webinar recording, as well as a PDF of a solutions brief. And we encourage you to forward it along to colleagues you believe that will also find it of interest. And with that, we'd like to open up the discussion. I think, Atife, you're the most popular question right now with the upvote. And the question is, when you say the modified version will be available forever, will it be available to all users in various companies or only users of Benchling in a specific company? Andrew, do you want to jump in?
Yeah, absolutely. That's a great question. So all of the data that is kept in Benchling is kept in a like customer by customer basis, and so um, whenever you set up a Benchling account, it actually is specific to your particular company. So we actually create and silo off, provision a separate server, essentially a separate tenant of Benchling, so that all of your data is kept separated from any other Benchling user. Though we are, a cloud platform, we do take data security and privacy very seriously, and make sure that all of our customers' data is properly partitioned and siloed away from any other customers.
Renee asks, where do you identify conjugations such as GAINA, C, TOCO, CHOL, etc.?
Yeah, that's a great question. There's definitely a lot of increase in usage of these different conjugates, such as GalNAc, on some of these oligo entities. The functionality that I showed today was primarily around storing and representing modifications that are on the sequence itself, on the oligo entity itself. Everything that I've shown was primarily on a particular nucleotide. So there's actually two downstream features that we're working on that would help enable both representing these chemical conjugations and even just different drug payloads, as well as linkers and et cetera. And so one is we're actually looking to add support for chemical entities in a kind of first-class way, which would be the correct data representation sort of way of keeping track of these chemical entities. And the second is we actually have another feature coming out very soon that is meant to help track what we're calling like terminal conjugations or terminal modifications to these oligos. And so we'll want to actually parse them out and store them as like linked entities or fields in schema fields.
I'm going to hop to continue with Renee's questions down. Does the syntax account for stereochemistry?
Yeah. And so within our monomer library, we actually take in a chemical structure representation of that particular monomer. And so we do have an ability to make monomers stereochemistry aware. It's just really going to be up to the customer, to the user, to basically provide that stereochemistry aware either mol file or SMILES, canonical SMILES string that represents that.
Greg has a question that might be a little bit of a fun tech challenge for us. On the fly, can you depict an aligned sense and antisense duplex?
Yeah, that's definitely a great question. What I've showed today is really foundational in that we are now able to store, modifications on a single strand of either DNA or RNA nucleotides. And so this, I think, also relates to another question we have down the bottom from Johan about supporting additional Helm data types like ADCs. One aspect of Helm that we really like is its flexibility to represent increasingly complex sort of biomolecules. And on the nucleotide side, this could manifest itself in different duplexes, such as like true siRNA duplexes or what have you. On the protein side, this manifests itself in either representing non-natural amino acids on a modified peptide string, or higher level complexes such as antibody drug complexes that might be composed of multiple protein chains as well as chemical conjugates. And so both of those aspects are on our roadmap and are things that we are very excited about and led to our decision to really adopt Helm under the hood here.
The next couple of questions from Johan and Renee together around our monomer library. Johanna asks, you mentioned collaboration. Oh, sorry, we just jumped around a little bit. Renee asked, does a user have access to specify any, well, we're all jumping around. Okay, so Johanna, as you mentioned collaboration with CROs, do you use the existing monomer.org, or is the monomer library a new one from Benchling?
Yeah, so the monomer library concept that I showed today is actually specific to a customer's Benchling instance or Benchling tenant. And so it can be configured to mirror monomer.org. We actually have an import tool currently to help directly import monomers from monomer.org. All of the monomers and modified nucleotides that I actually showed today, I just imported directly from monomer.org. We do have a way to do this internally and on the Benchling kind of admin side, and we're going to be looking to expose this to our users over time and build out a UI to allow them to directly create, import, and curate their monomer library. So definitely not very aware of monomer.org and not looking to formalize a new concept necessarily.
And then Renee's question, I think, is really related. Do users have access to specify any unique monomers made in-house in the database?
Mm-hmm. Yeah, so we definitely have the flexibility to specify unique custom monomers made in-house. We have not fully exposed the UI path to do this yet, but we have internal tools to help us basically curate and manage all of the monomers that might exist in a customer's monomer library. Again, as long as a customer is able to provide that chemical structure representation of what that monomer is and provide some of the additional details, the Helm details around the attachment sites, the specific natural analog that monomer is supposed to correlate with, we can customize your monomer library as you see fit.
I'm going to hop down to Nick's question just because it's the same vein of topic. If an oligo monomer is registered, can the sequence history still be accessed and reverted to?
Yes, absolutely. It doesn't matter if an entity is registered or unregistered. That entity and all data associated to it, regardless of if it's associated results data or analytical data, or it is actual sequence history data, all of that continues to persist with that same oligo entity, regardless of its registration status. And so kind of gets back to an earlier comment about preserving data integrity, right?
Swapnil asks, does the platform calculate properties of duplexes such as extinction coefficients, mol.wt, conjugation, site, etc.?
Yeah, that's a good one. And so with the computed fields that I showed for actually computing the Helm in IDT notation, we have a few ways to actually implement custom computation with computed fields as well. So we do have some ways of calculating, extinction coefficient and molecular weight using computed fields. The actual computations, especially for custom monomers, might need to be configured, but we can definitely work with each of our customers to define the particular algorithm, the particular parameters or constants needed to accurately calculate any of those properties.
Renee asks, can sequence map expand into full molecular view?
Yeah, that's a good one as well. So we are tracking now the chemical structure of each of the monomers used in a nucleotide. And so that particular full atomic or full molecular structure sort of view is another downstream roadmap item for us. The real goal here is to use Helm and the structured way of breaking up sequence entities into chemical structures as a way to help translate entities that could either be represented fully as a sequence string or fully as a chemical structure. And so in this case, being able to actually render and combine all of the chemical structure of those monomers together in an intuitive way. So.
Great. Jack asks, after designing the modified RNA DNA oligos, where can we order to synthesize these molecules? And what is the methodology for validation of its efficiency?
Yeah, that's a great question. We will allow the export of these, modified oligo entities from Benchling. We currently support, that natural-based syntax, the Helm syntax, and certain vendor syntaxes like IDT. And so in cases where you need to export those as a particular vendor syntax and send an order to a vendor, you should have the available resources there to do such. We're also looking at down the line, adding out support for additional vendor syntaxes. So Benchling can serve as that centralized hub to help you translate or change syntaxes in cases where you want to actually send some orders to like a gen script or a Dharmakon.
While we're talking syntaxes, Fred has a real-time question for an error state. And we might, I think, probably also follow up directly, Fred, but he's trying to make a modified RNA using Helm notation, and he gets this error. Only Helm version 2.0 syntax is supported.
Yeah, so we do need to configure and enable certain customer tenants to accept Helm syntax, and would love to follow up with you after the webinar on in this particular case. It could just be a case of the tenant just needing to be enabled, but can follow up after.
Nick has a couple questions for us. Is there a timeline for the public implementation of conjugation additions to oligomers, and any expectation as to when the users can take advantage of it?
Yeah, so this is actually in flight and almost ready for public access now. And so, conservatively, I would say we're a month away, but realistically, probably shorter than that. So would definitely like to follow up with you offline too, and can let you know when we do release those capabilities.
So stay tuned, huh?
Yeah.
And then he was also following off of Jack's bio question. Is there any internal ordering capability between Benchling users?
We don't currently have an integration with different synthesis vendors to initiate orders, but that is actually a hot topic for us, and a project that we're really looking to invest in over the next few quarters. So definitely stay tuned. I think there's quite a lot of interest and demand, not just from folks attending this webinar, but across all of our customers for a better integrated solution with all of the most common synthesis vendors. So
I think with that, we've wrapped all of the questions. Again, please reach out to Andrew and myself directly if you want to continue the conversation, as well as visit our website. We want to thank everybody for attending live today, and look forward to continuing the conversation wherever possible. Thanks so much. Have a great one.